Okay, welcome to the third part of chapter 6 of the chapter 6 lecture. And so in the previous part, we had talked about different ways that we can manipulate the nutrient and environmental requirements of microorganisms to be able to culture and study them in the lab. And there's many more examples that we'll use in the lab specifically, so you'll have a lot of ways of interacting with microorganisms in the lab. Now what we want to do is talk about the phases of microbial growth. And so I'm going to start by reminding you of binary fission, which is kind of the predominant way that microorganisms tend to grow and reproduce. And then we'll talk about how kind of those phases play out as a population is growing in the lab.
Bacterial cells do not divide by meiosis and mitosis. Instead, most bacteria grow asexually by a process known as binary fission. In this process, the bacterium copies its chromosome, elongates, and splits off into two daughter bacteria with a genetic makeup identical to the parental bacterium. Each daughter cell can continue the doubling process. This process happens rapidly, so the number of bacteria present in a solution can increase very quickly.
Okay. So when we're isolating microorganisms and culturing them in the lab, we often start off with just a very small aliquot of those microorganisms. For example, if you're making an environmental plate that you just leave open in the lab, you just have a couple of bacteria that land, or a couple of fungi that land on that plate from the open air. Or maybe you intentionally are culturing some microorganisms from one colony to another plate. And so you're just transferring over some of those microorganisms, right? And then when they land on that media that is nutrient rich and it has the correct conditions for their growth, then they are going to start to grow and reproduce. And when we say grow, we're often talking about the population growing. And so the individual cell will, duplicate its chromosomal DNA and continue to divide. And each cell will divide as it is in that media. And so instead of just adding members of the population on kind of arithmetically, we actually add them exponentially. Because instead of parents kind of then producing another organism over time, another offspring over time, you have each individual daughter cell then divide. Each daughter cell divides. And so we have exponential growth. And so it's pretty typical for the process of binary fission to take about 20 to 30 minutes, which means in the first couple hours, you'll have a cell division into 2, and then 4, and then 8, and then 16, and then 36. And in a couple hours, you have about 64 cells. And then only about 7 hours later, you'll have millions of bacteria, because at each interval, you're just duplicating the number of organisms that you have. And this is why we don't leave foods out, for example, because when you leave something like chicken out of the refrigerator, what you've done is taken microorganisms that are on the chicken out of the refrigerator, which means that now they're in a temperature that is more optimal for their growth, whereas the refrigerator inhibits their growth, or the freezer inhibits their growth. And then they start dividing because they have the nutrients, which are the chicken in this example, to grow. And they divide exponentially and quickly. And so one salmonella or a couple salmonella cells, which are actually very typical that you have salmonella on chicken from the store. In my previous lab, we used to test chicken from the store, and about 50% of the samples have some salmonella on them. Pretty common. And that's why we cook it. Refrigerate, freeze, cook, all these things. A couple of cells of salmonella very quickly become millions of salmonella cells. And then that can make you sick, right? Whereas your body might be able to address 1 or 2 cells, but millions of them, that's going to overwhelm their immune system, right? And so it's because of that exponential growth that we see this. And so what does that look like? Well, Exponential growth, when you see doubling of your population at each interval, so like we said, a very common interval is 20 to 30 minutes, you're seeing a doubling of the population. It's different than like if you had a group of, mammals, for example, and you see their population increase kind of over time. Although even mammals can have exponential population growth if they have all the resources that they need. So that's a caveat too. But bacteria they double their population at each generation interval, and you see this very quick exponential growth. Okay. Now, just like we mentioned with mammals a second ago, nutrients are important. So even though bacteria can reproduce very, very quickly, at some point, whether it's in an environmental setting or in the lab, whether it's because of competition or the media then is, depleted of nutrients, at one point, you're going to see a plateau of the growth of those organisms. Very similar to what we see when we talk about ecosystem biology and we talk about carrying capacity, right? So those organisms are kind of start to max out on that media or that nutrient, and they're going to start to plateau. Not all of them are going to be able to divide and reproduce. And as a population, the whole population is kind of start to stabilize. And at one point, there's going to be significant competition, and you'll actually see death or decline. And so we have to be really thoughtful about this in the lab. We tend to want to see organisms around their exponential or at least beginning of their stationary phase. This is when they tend to be most active. If there's an enzyme that you're looking for, it's going to maybe be more likely to see it in these phases. Generally speaking, although there are some tests that we want to do in different phases. So for example, we've talked about endospores a lot in this class. Endospores are when you have certain species of organisms that are able to produce endospores, and they will produce them when they feel that the environment is no longer kind of encouraging or optimal for their growth. They think they're going to die. And so when we do our endospore stain, we actually want organisms that are in a death phase. And so we actually use cultures that are old on purpose because they're more likely to have endospores in them, right? Whereas if, for example, we want to keep a culture for a long time, we want to make sure that they don't move into a death phase. So we want to make sure that we optimize the amount of nutrients that are present. We make sure that the airflow is correct so that media doesn't dry out and so that they can actually exist for a long period of time on that media. So in that case, we don't use plates, which can dry out very quickly. Instead, we use broths or slants, okay? There are cases in which we actually want to grow cultures over very long periods of time, like months. In that case, we either need to reculture them, right? So as the media kind of is depleted, we then plate them on a new set of media and continue to reculture, or we actually have to have a continuous addition of nutrients into that media. And so that usually looks like a liquid broth that has a continuous introduction of new media. So as the nutrients in that media get depleted, then that media is kind of released, and then new media is added, and the organisms can continue to grow over long periods of time. So in the lab, we won't do this chemostat method where we actually add nutrients. We'll just continue to reculture our cultures onto new plates so that they're continuously having a fresh kind of nutrient supply, and they'll stay alive. Now, think of our lab term by term. So over the summer, we don't actually reculture our plates. So how are those cultures being stored long term? Or maybe, for example, HeLa cells. So HeLa cells, very interesting conversation around HeLa cells and lots of equity, issues with that, racial equity, gender equity, lots of issues. And so look to the book actually written by a PCC alum called The Immortal Life of Henrietta Lacks. But think about cells like that are decades old, right? And so you need to keep those cells for long, long periods of time. We do have mechanisms for freezing and freeze drying those cells for storage over long periods of time. Okay. And there's no really images associated with that, but you essentially would freeze them or freeze dry them, and you would keep them over long periods of time. And then whenever you needed some of those cells, you would go into those freeze dried cells and then re-incubate them onto media in the correct kind of physical environment, and they will essentially kind of come out of dormancy and continue to grow. Okay. And so in that way, we're able to culture many different types of organisms, many different types of media, maintain them over long periods of time, weeks, months, and even decades, to be able to test and kind of understand them. Okay. It's that discussion around kind of bioethics and equity in science is really interesting and really essential. So if you have interest in that, I'm also going to include a link below to the book, The Immortal Life of Henrietta Lacks. It's a very good read, and it really opens your eyes to a lot of that complexity, since we mentioned it. Okay. The last thing that I want to talk about is, as we're growing microbial cultures, we might want to check in and see how those cultures are doing, so how many of them are surviving. Or we might want to look at a food sample, for example, and see how many microorganisms are on that food sample, if there's a significant number of pathogens, for example. And so another really important method that we use in the lab is cell counting or cell enumeration methods. This is a cell enumeration method that allows us to use a microscope slide to count the number of organisms in a culture. This is not something that we do in our lab specifically. Instead, we use what's called a serial dilution. And the serial dilution allows us to take our sample. So we'll use toothbrushes, sometimes we use cosmetics or food items, and then you can actually make a serial dilution off of that item and then plate and grow the organisms from all of those dilutions. And so what you expect is that the original sample will be, if you consider, or if you expect to be contaminated, will grow a lot of microorganisms. But then as you dilute out, there should be a plate with a countable number of microorganisms, and then you can calculate back and understand how many microorganisms were in your original sample. And so there's different ways of enumerating microorganisms in samples. This is the one we'll use in our lab, although there are other ones like this microscope slide that is mentioned in your book as well. Okay. So that's essentially chapter 6 for us. And what I want you to do for your discussion post for chapter 6 is take these different components, right? And say, okay, at the end of the chapter, we talked about the phases of cell growth, right? We have an exponential phase, a stationary phase, and a death phase. And those are going to be dependent on the characteristics of the nutrients that have been supplied and the kind of environmental characteristics around that microorganism. And so think about how you might change the nutrients, the pH, the temperature, oxygen levels, community interactions, as we had talked about before, and how that might impact the growth of the microorganism. You don't have to talk about all of these. You can focus on one or two. And under what circumstances within that category would growth be positively impacted or negatively impacted, and for whom? Okay. And so there's lots of examples in our chapter today that kind of give us some insight into how you can change different components of the media or different components of the environment, or different kind of environmental factors, and that will impact the growth of an organism. It might impact the whole community, or it might impact specific organisms within the community. And so start to have that discussion in the chapter 6 discussion board. Feel free to focus in on specific nutrient requirements or environmental conditions, or feel free to focus in on specific types of microorganisms. Like I want to talk about halophiles specifically, or I want to talk about these organisms that live on glaciers in really cold environments, so on and so forth. So you're welcome to specify and kind of discuss how, if you change that environmental factor, how it'll impact the growth of that microorganism. So thank you for being here for chapter 6. And we're talking about nutrient and environmental requirements as the first part. Definitely recommend you have kind of a flow chart that summarizes that content. Then the second part, we talked about how we manipulate these nutrient and environmental requirements so we can grow specific microorganisms or learn about characteristics of microorganisms in the lab. And in the third part, we talked about the growth phases of microorganisms and how we can kind of learn from those phases. And also we talked about enumeration in that third part. And so I look forward to reading your discussion posts, and I will see you in the next video. Take care. Thanks.